And I feel for all of you that have alpha-gal syndrome. I do not have it, but I have read many, many papers. I've talked to many patients, and it is a debilitating condition you have. I was speaking to somebody earlier today, and the social aspect of having alpha-gal syndrome is horrible. Difficulty going to visit friends, to go to a restaurant and eat. My colleague and I, Dave Ayers, we're actively trying to find some solutions to heal you, to make life better for you. And if you have any comments about what we've done, we'd love to talk to you afterwards. Just so we're on the, who is Revivicor and what have we done? Revivicor is an animal biotechnology company based in Blacksburg, Virginia, and we trace our origins back to Dolly the sheep. One time we were a subsidiary of PPL Therapeutics, and the researchers at Revivicor and in Scotland worked to clone Dolly the sheep. Revivicor was the first to clone a cow. We were the first to clone pigs. And we have the first to establish a line of pigs that is alpha-gal null. Dave, my colleague, will be talking more about this pig later in the latter half of the presentation. To make sure we're all on the same page, and you guys know this story well, this is an article, an image from an article published by Dr. Commons. And what's really interesting, this article was published in 2015, and the map of the Lone Star tick is from 2015, and you may not notice it, but the range is just north of Virginia. But if you compare that to the map that Jennifer showed in her presentation, it stretches all the way up to the Great Lakes. So in 5 years, the CDC, or 4 years, the CDC has changed the range of the Lone Star tick to encompass quite a bit more of the country in just a short period of time. But the tick is the carrier, the vector of transmission, as Jennifer put it, carried around by all the deer. And in Blacksburg, where I live, my wife and I are confronted with deer all the time in our neighborhood or just around our house. The tick bites you. You develop certain patients, such as yourself, develop anti-gal IgE, and there's one lab in the country that runs that test, Eurofins, Viracor, and you can get it done through your physician. And subsequently, after the IgE levels go up and you're exposed to alpha-gal through mammalian food products, you develop an adverse event, which often includes anaphylaxis. You'll see in the talk that I have, it's more than food. There are much more dangerous things out there than food. This is somewhat of hopefully it's well understood by this audience, but there is a method of going through and doing a risk analysis. I'm not going to do a quantitative risk analysis. It's going to be a qualitative risk assessment where we identify the hazards and the exposures, and then multiplying those two together gives you a relative idea of what the risk is. So when we look at it, what the hazards are, the prevalence of alpha-gal. The alpha-gal is in all food-producing mammals, and it's in all the derivatives of those food-producing animals. And sometimes those derivatives are obvious, and sometimes they're not so obvious. The arachnids, as the vector of sensitization, they're the ones that drive up these anti-gal IgE values. The prevalence of anti-gal IgE antibodies in the human population. Interestingly, all of us have anti-gal IgG and anti-gal IgM, but this is not responsible. These anti-immunoglobulins are not responsible for anaphylaxis. It's the anti-gal IgE that's responsible for anaphylaxis. We are in an incipient phase of knowledge. I've talked to patients that have had this condition for more than 10 years, and they had to figure things out on their own. Since the research has come out at the University of Virginia, some healthcare providers have figured this thing out, but I would not solely point the finger at the healthcare providers for not understanding. There are others, and this includes the regulatory authorities, and Dave and I have hit the FDA over the head with this thing. And it's the product manufacturers. Dave and I have talked to many folks who are responsible for putting together food products and for medical products, and it falls on deaf ears. And I implore you to have your voice heard. Help Beth, help Jennifer. Your voice needs to be heard, not just by the healthcare providers, but your legislators. Something has got to be done about this. One of the other major hazards is there's no warning labels. Someone asked the question about where does this alpha-gal come from? And it comes from a gene that is on mammals, and that gene is the GGTA1 gene. And it's active, and it's the one that's responsible for the expression of gal. Remarkably, 28 million years ago, the gene in the humans and old world monkeys was inactivated. There was a mutation done, and I won't go into the details, but that mutation was very valuable to our immune system. And it's part of the reason why when you're exposed to gal, you react vigorously. There is a set of species that do not have an active GGTA1 gene, and those are the animals that you can eat, and that's birds, frogs, and fish. So birds, frogs, and fish do not produce alpha-gal because they don't have the gene. The prevalence of alpha-gal, so all food-producing mammals have alpha-gal. It doesn't matter if they're organically raised, if they're free-range, they have alpha-gal. Food products, the obvious food products are a great poster out there that made this point. Obvious things like meat, but then there's not so obvious things like pork rinds and gelatin and things like that. It's in a lot of mammalian products are used for active ingredients in drugs or excipients, including vaccines. It's in cosmetic products, and it's also in medical devices. Lisa highlighted the heart valve, and I'll talk a little bit more about that later in the presentation. And the arachnids, and once again, here's the map of the Lone Star tick, and this is taken from 2019, and you can see it stretches all the way up to the Great Lakes. Jennifer mentioned the black-legged tick is implicated, and chiggers have been implicated as well, but in the article it does indicate that there could be some confusion between a chigger and a nymph of a Lone Star tick. Also, the chiggers have not been evaluated whether or not they synthesize alpha-gal. Jennifer mentioned the Asian longhorn tick, and this map on the bottom is indicating where its range is expected to spread. And it's not a big problem right now, but I fully expect it to be a huge problem, and it has been implicated in sensitization of alpha-gal. There are other arachnids around the world that cause alpha-gal. A lot of research has been done in the country of Sweden, Germany, some in Spain, and a lot of work done in Australia. In fact, the group in Australia and the group in Virginia share in the discovery of alpha-gal syndrome. This is a map of sort of the prevalence rates, the percent of people who are positive for alpha-gal. And the picture of the United States is taken directly from the USDA's prevalence data. It is remarkable. remarkably high, 37% estimated to be in the region of the country where we are right now, 34% in the southeast. And then when you move over to the European community, you start to see similar numbers, maybe not as high. Sweden, 10%. Germany. This paper, I pulled the information out on Germany. They did a random survey looking at people who were outdoors, spent a lot of time outdoors. This would be hunters, forest service workers, and their prevalence rates were twice the population of people who did not spend a lot of time outdoors. Somewhat obvious, I guess. If you're around ticks, you're going to get bit by them. Just want to highlight Italy. This paper, they surveyed almost an entire town, a random population, and in this rural area, and the prevalence rates were 25%. I want to just emphasize, just because you're allergen positive does not necessarily mean you'll react if you consume mussel. So it's a much smaller population that actually is reactive to mussel. It's probably about 10% of the allergen positive population. So that leaves probably 90% of the people that are allergen positive that probably don't even know that they have the potential for a reaction. And that is going to be an important point when I show a few slides later. I talked about the incipient phase of knowledge where we are right now, and the discovery was in 2008, and if you were just diagnosed recently, you should feel somewhat good about it because there's knowledge out there that there are healthcare providers. You may have to look a while before you find them, but they are out there, and there is information out there. Unfortunately, Facebook may not be the best, but I know Facebook has a lot of good communities and a lot of interesting information out here. But this plot is one that we created at Revivicor, and it tracks the number of scientific publications that have been published over the years. And you can see it is increasing, and it's been a 78% increase between 2014 and the May of 2019. As far as the incipient phase of knowledge, the NIH recently, great thing, National Institute of Health just recently published a paper describing the allergy. It is an uncharacteristic allergy, very, very different from all the other allergies that are out there. Paper published from a group in North Carolina where they looked at a population of patients that had been diagnosed, and they went and interviewed them and tried to understand their history, how they were diagnosed. And maybe not too surprising to this group here, 9% of the time they got a correct diagnosis. Remarkably, from the time of their first event to the time of proper diagnosis was 7 years. That's ridiculous. 7 years you have to navigate the healthcare profession before you get a diagnosis. The patients often telling you, in some ways I can understand the healthcare provider. I read it on the internet, I heard it on the radio, I read it in the newspaper, and the healthcare provider dismisses them. The patient goes in front to go talk to the healthcare provider, can you run this allergy test for me? And the healthcare provider pushes them away. It's an incipient phase of knowledge for the regulatory authorities. Several articles where allergists are demanding warning labels on drug products. Surgeons, remarkably, surgeons writing letters to the editor demanding that alpha-gal get removed from heart valves, and they're employing regulatory scrutiny. There are no warning labels. The Food and Labeling and Consumer Protection Act recognizes 8 major allergens. Alpha-gal is not one of them. I live in, is anybody here from Arkansas? Arkansas. Congratulations, Arkansas. By legislative action, they have established the Alpha-Gal Task Force. And that task force is commissioned, mandated to do two things. Educate first responders to go, so they know when they find somebody in anaphylaxis to think Alpha-Gal. And the second was to petition the United States Congress and the FDA to change the food labeling laws to include Alpha-Gal. They were unsuccessful. There's been no change to the law. The country of Japan, I recently read, for one of the drug products, cetuximab, and that could be a lecture in itself, Lisa, to have somebody come in and lecture on cetuximab. It is an awesome story. It involves Martha Stewart going to jail, and it also involves Alpha-Gal. So maybe put that on the agenda. If anybody wants to know, I'd be happy to talk to you, and I'm sure Beth and Jennifer can relay the story as well. But anyway, the drug cetuximab contains Alpha-Gal, and the discovery of Alpha-Gal syndrome is directly linked to cetuximab administration in the United States. But the country of Japan mandates on the warning label for this drug that people who are recently bitten by tick bites it would be contraindicated to administer this drug. So congratulations to Japan for staying on top of this. It's not all doom and gloom, and there is a lot of knowledge and awareness that is increasing. It's wonderful to watch, from my perspective, the different activity that's just occurred in the past year. Jennifer mentioned Commissioner Troxler's Food Safety Forum, and it's going to be an all-day event on Wednesday, this day dedicated to Alpha-Gal. Lisa, this is your second symposia, and I kicking myself for missing the first one. So hopefully we can be included in all the subsequent symposia you hold. The CDC just started tracking Alpha-Gal. Beth's going to talk about the tick-borne working group, and I just want to say one thing about it. The first report went to Congress, and it was a 109-page report, and I was quite disappointed that only one page was dedicated to Alpha-Gal syndrome. But then I went in and read the patient comments, and it's overwhelming the number of Alpha-Gal syndrome patients that wrote letters to the working group. And so I would encourage all of you to be active and write letters. I was quite moved, Dave was quite moved, and I've used those letters in many of the internal discussions that I've had at Revivicor. Morgan Griffith is a congressman from Virginia, and he highlighted Alpha-Gal syndrome in his newsletter in April. And I mentioned before, the National Institute of Health has published a letter, and they also are developing a working group. So exposure as another part of the risk analysis, and every one of these exposure methods has been documented in the literature, and this can include inhalation. I don't know how many of you avoid walking into the meat department in the grocery store, or can't stand cooking a bacon, or have to leave a house. But it's been documented. Not a lot of papers on that, but it has been documented. External application, where you're applying things to your skin and you have an adverse event. Consumption, not just of food, but of drug products. And those drug products can include suppositories, implantation of heart valves or other medical products, and injection. And injection probably is the most hazardous thing. You're bypassing the digestive system, and you're putting Alpha-Gal directly into the immune system. So lots of papers published on consumption. These three highlight one in the United States, Dr. Commons. He's one of the world authorities who was at UVA and is now at the University of North Carolina. Dr. Van Nunn in Australia, she's published extensively on it, and the group in Sweden has published extensively on it. One thing, particularly for this group, that you might want to be aware of is these are immunohisto stains, and they are tagging Alpha-Gal. So this is a piece of pig muscle, a piece of pig kidney, and a piece of pig liver. And all that brown is alpha-gal. And when you first look at it, you have to look really closely at that muscle. There's hardly any alpha-gal in the muscle compared to the other tissue types. But that's enough to put somebody in the hospital, send somebody to the ER. Kidney, it's a little more heavily concentrated, and then liver, I'm not sure you would survive consumption of liver. Maybe that's a good reason to avoid it. I ate liver once. I don't think I'll ever eat it again. But in European countries, kidney is a delicacy, and it is eaten. And this concentration is commensurate to the clinical presentation. So when you dig down and you look into the literature, I mentioned the German paper. They identified 10% of their population that was reactive to muscle meat. A pretty small percentage, you might think, but if you carry that out into the bigger world, that's probably comparable to any other allergen in the that's recognized by the law. But when you move to something like kidney, 50% of the patients will react, and it seems kind of obvious. There's more alpha-gal in the kidney than in the muscle. And when you move to liver, I haven't seen anything published in the literature, and I really wonder if that's because those patients don't survive if they eat it. Cetuximab, when you move away from consumption and into something like an intravenous exposure where you're putting the drug directly into the bloodstream, the original paper, 23% of the patients that received cetuximab, 18 of the 76 were identified to be allergen positive, and 18 of those patients reacted adversely. And some of that adverse reaction included death. So when you move from consumption to a drug that's intravenously rejected, it's a much, much greater hazard. There are other drugs. So vaccinations, I encourage everyone to get their children vaccinated. Our children have been vaccinated, but there has been papers published where I can't imagine being a parent of this, but went in to get their child their booster shots of measles, mumps, and rubella, and there's gelatin in the vaccine, and upon injection of that vaccine, that child went into immediate anaphylaxis and had to be rushed to the ER. It just shouldn't happen. There's no reason to have alpha-gal in a product to a susceptible population. The heart valves. Heart valves are a unique and special case because those things are designed to last a long period of time, 15 years. Well, the case study, and this included Mr. Reynolds, they got the heart valve implanted, and they're walking around and they're doing great. 5 years later, 5 years post-operatively, they decide to go for a hike in the woods and get bit by a tick, and within months, they're going to the ER because they've had a hamburger, and they've gone into anaphylaxis. And within 6 months, that heart valve is acutely rejected. It's no longer functional. Mr. Reynolds had some special care to prevent, to get his reoperation done and get the proper valve in, but I often wonder how many people are not being captured. There's only one report in the literature. Dave and I have had the opportunity to speak to 4 cardiovascular surgeons. 3 of them practice in the Southeast, and when we described this situation to them, they looked at us like we were from Mars. What are you talking about? One of them has engaged in the story and actually is developing his own research program. Moving to some other products,
This is, once again, these histologies are from certain pig tissues. The one on the top is pancreas and thyroid from the pig, and the intestines and the lung. And I think you can see various, the brown staining is where the alpha-gal is, and I think you can see that some tissues like lung are pretty heavily concentrated, even the pancreas. And what you see next to them is the pancreatic enzymes. Those are derived directly from a pig pancreas. The thyroid medicines, there are thyroid enzymes that come directly from a pig. And there's heparin. Heparin is used quite a bit in surgery for medical applications, and it is derived from lungs and intestines from animals. And there's not a whole lot published about these drugs, but there are a few things that are in the literature. And at Revivicor, we've done our beginning to do our own sort of research on these products to develop this story better. But in the pancreatic enzymes, those drugs that are out there are more potent than a pig kidney. There is more alpha-gal in that. I'm not sure a patient would survive one pill of pancreatic enzymes. Likewise with the thyroid enzymes, there's quite a bit. It's work that we've developed at Revivicor, and it was not quite ready for prime time, but we've looked at these thyroid medicines, and it looks like there's quite a bit of alpha-gal in it. And heparin, it has been documented in the literature when these patients were administered heparin perioperatively that they started to develop signs of anaphylaxis before they intervened and halted that process. So in our qualitative risk assessment, there's substantial hazards out there, and the exposures are quite a bit. And this leads to the conclusion that there's a high likelihood of adverse events due to alpha-gal. And there is some data in the literature to back this up, and this was just published last year, and it was a group in Tennessee. Once again, they're in the epicenter of the Lodin Star tick, and they retrospectively reviewed 10 years worth of patients who were admitted to the hospital for anaphylactic shock. So this is the most extreme form of anaphylaxis where it required admission to the hospital, and they identified all the triggers, and alpha-gal was the number one. It was the top of the list, and kind of remarkably about it was when I first read this, that it's just shock, but all other foods were identified as number two. So they do list medicines, but they didn't say what medicines, so there may have been alpha-gal in those medicines. And they also list exercise, which exercise is a cofactor. So if you decide to eat some meat, don't go out and run 5 miles. With that, I'm going to turn it over to my colleague Dave Ayers to talk about our unique product.